Have we all assembled here? Dr. Willerson, I see, is here. Historically, a historical anecdote is that, as many of you know, our very first World Stem Cell Summit was in Texas at Baylor College of Medicine at the Texas Medical Center. And not only did we have Dr. DiFede give the first talk ever to our summit, we were very honored to have a wonderful scientific presentation by Dr. James Willerson. I'm honored to have you. Who knew? 10 years, here we are again. You haven't changed a bit. I'm just honored to have you here, and it's very meaningful for 10 years in the journey. So I'm going to introduce Darcy DiFede from the University of Miami, who has I work very closely with at the University of Miami Miller School of Medicine, the Ischaemia Institute of Stem Cell Research there, and I'm on a governance advisory board, and as a graduate, a double graduate at the University of Miami, it's good to see my alma mater is investing so heavily in this field, and especially in the cardiovascular space. So Darcy, thank you for moderating this session.
Good morning, everyone. Welcome to the cardiovascular clinical trials. This is the area that would benefit most due to a huge unmet need today. The regenerative field is showing great promise in this area through advancement of clinical trials. One of the most important mechanisms in achieving this is funding from the NIH for clinical trials to advance the field. In this, the CCTRN, which is Cardiovascular Cell Therapy Research Network, is funded by the NHLBI. Both of our speakers today are principal investigators within the network, and it is a privilege to work with them. Our first speaker is Dr. John Cooke, who is currently the chair of the Department of Cardiovascular Sciences and director for the Center for Cardiovascular Regeneration at the Methodist Hospital Research Institute. Dr. Cooke served as faculty at Harvard and professor of medicine at Stanford for over 20 years. He serves as principal investigator for the Cardiovascular Cellular Therapy Research Network, funded through the NHLBI. He ranks among the top in his field as a clinical trialist and basic scientist. Dr. Cooke.
Thank you, Darcy, for that kind introduction, and to Mr. Siegel for putting on such a fine meeting. It's been just wonderful. I've personally learned a lot at this meeting, and I look forward to coming again next year. I'm going to talk about the a cardiovascular disease that's often ignored or not well studied, not well recognized, and that is peripheral arterial disease, which is due to the same mechanisms that cause coronary artery disease. Atherosclerosis is the major cause of peripheral arterial disease. Atherosclerotic arterial occlusive disease affecting the vessels that supply blood flow to the lower extremities. And this disease affects over 8 million Americans, and it typically is something that results in pain with walking, but can lead to gangrene in later stages. We do have good medical therapy to reduce the risks of morbidity and mortality in these individuals, the cardiovascular mortality, by reducing the risk factors. So many of the same drugs that we use for our patients with coronary artery disease, we use for these patients with peripheral arterial disease. We use statins, ACE inhibitors, angiotensin receptor blockers. We use tobacco cessation, antiplatelet agents, metformin. And these do result in a reduction in mortality from stroke and heart attack. The other thing that you want to do with these individuals is you want to relieve their symptoms, and their symptoms are typically related to use of the legs, so they have difficulty walking. Our medical therapies are not as good. We only have one approved drug that's really useful, that's Elastisol, and it improves walking distance by about 30%. Actually, a good supervised exercise program works much better, can improve walking distance by about 100%, can double one's walking distance. And then in occasional patients, we do need to make interventions. We need to do surgery or endovascular intervention to open up the occlusions. What about stem cell therapy for these patients with peripheral arterial disease? What is known? Well, there have been over 50 clinical trials in peripheral arterial disease of various stem cells, and I've summarized the literature recently. In this table, you can see the trials that have been done, and there are promising results of these trials, somewhat encouraging. Most of the trials were done in patients with critical limb ischemia. I'll differentiate between that and intermittent claudication in a moment. Most of the trials were done using bone marrow-derived mononuclear cells, and the caveats with these trials, although some of the results were encouraging, some of the surrogate endpoints were encouraging, many of the primary endpoints were not achieved. And in summary, after a lot of trials, over 50 trials, and over 15 years of work in this area, we still really don't know what is the right cell, what is the right method of delivery, a lot of unanswered questions. And I've listed here some of those questions for you. As in all fields where we're using cellular therapies, what type of cell is the optimal cell type? For this disease, we still don't know. There's a move now toward allergenic mesenchymal stem cells, away from bone marrow-derived mononuclear cells. Should we be using combination of cells, or should we be enhancing the cell therapy in some way? As you've heard at this meeting, our patients with cardiovascular disease have demonstrably impaired stem cell function. They have fewer endothelial progenitor cells, for example, circulating in the blood. Those endothelial progenitor cells that are available in our patients with cardiovascular disease are demonstrably impaired. Their replicative capacity is reduced. Their ability to integrate into a vascular network is reduced. That has been demonstrated. The more risk factors that you have, cardiovascular risk factors, the more impairment there is. So perhaps we should be doing something to resuscitate the stem cells before they're injected. And we heard a nice talk from Dr. Atta at the Mayo Clinic and his approach to reviving stem cell therapies for cardiovascular disease. Let me help you differentiate between these two disorders of critical limb ischemia and intermittent claudication. The question here is which target is best for stem cell therapy? For critical limb ischemia, patients with gangrene, with pain at rest, these individuals have severe peripheral arterial disease, and as shown, they're easily identified. And they typically have disease in multiple segments of the vasculature. They typically are not amenable to endovascular intervention, surgical intervention, often fails. So we desperately need something new. The medical therapies for these patients are not very good. The surgical endovascular therapies can fail. So we need something for these individuals. So another reason for stem cell therapy, good target. On the con side, these are difficult patients. They're very difficult. They often have other comorbidities that are severe, like renal failure. These individuals have high mortality and morbidity. Over a period of about 2 years, 20% of these individuals will succumb to cardiovascular disease. So it's a difficult target in that respect. That's one reason perhaps to go for after lesser severity of the disease. Patients that have lesser severity of peripheral arterial disease will manifest with difficulty walking. They'll get pain in their calves and thighs, buttocks with walking. And those individuals are also potentially a target for stem cell therapy. Because again, the medical therapies that we have are not ideal. There's also a lot of these individuals, 8.5 million patients, so it's a large market. Another reason why you might want to use this clinical target for stem cell therapy. The other thing that makes these patients more amenable to a therapeutic approach is the regulatory roadmap is simpler. One can simply improve walking distance in these patients as a primary endpoint. You can do this using treadmill exercise testing or 6-minute walk, and the FDA will accept an improvement in walking distance for approval of a new therapy for these individuals. That being said, a lot of pharmaceutical companies have broken their wheel on intermittent claudication. There have been a lot of therapies that have been tried and that have failed. Vasodilator therapies have not worked for this condition. The only 2 drugs that are known to improve walking distance are cilostazol, which I mentioned already, which is primarily approved for that purpose. Interestingly enough, statins. Statins improve walking distance about 30% as well. Of course, statins we use to lower cholesterol and to reduce morbidity and mortality from cardiovascular disease, but they do also appear to improve walking distance in these individuals. One reason the vasodilator therapies do not work for these individuals is that this is a complex disease. It's not just vascular insufficiency. By the time these individuals come to recognition of their physician, they have a number of other problems in the lower extremity. They have metabolic mitochondrial abnormalities. They have neurological damage, nerve damage, and they have muscular abnormalities. They have fatty fibrous replacement of the skeletal muscle. So it's a complex disease. If we're going to use stem cell therapy in these individuals, what's the right approach? Should we be targeting the parenchyma? Should we be targeting the skeletal muscle itself? Or should we be targeting the vessels? A parenchymal approach makes sense because stem cell therapies can have immunomodulatory effects, growth factor effects. They can preserve the viability of tissues, and you can enhance angiogenesis, the microvasculature. But these individuals have long segments of occluded vessels, and they need more blood flow. Angiogenesis will not be sufficient to improve blood flow to these limbs. You need larger vessels. Arteriogenesis, the enhancement of existing collaterals, may be another approach, and stem cell therapies could be potentially used, and certainly preclinical studies have suggested that stem cells of various kinds, bone marrow-derived mononuclear cells, mesenchymal stem cells, can secrete factors that enhance collateralization. Just briefly mention a CCTRN-sponsored study that's ongoing. Darcy mentioned to you that Dr. Willerson and I are both members of the Cardiovascular Cell Therapy Research Network, funded by the National Heart, Lung, Blood Institute for clinical trials in stem cell therapies for cardiovascular disease. PACE is one of our studies, an NHLBI-funded multi-center clinical trial, and we are using Aldebright cells in this study. Aldebright cells enhance angiogenesis, and they are largely CD34 positive cells, CD133, also CD105. They're MSCs and EPCs, a combination of those cell types. And they're isolated by this Aldebright, which is responsive to the increased aldehyde dehydrogenase activity of these stem cells. The patients that are included in this study are intermittent claudicants. These are patients that are having symptoms walking, and their age should be greater than 40, typical atherosclerotic arterial occlusive disease. And we look for individuals that have a reduction in the blood pressure at the ankle, an ankle brachial index of 0.9. The injections that are going to be of the Aldebright cells will be done in this manner. It's kind of a hybrid approach. We're mainly targeting the parenchyma, the skeletal muscle, but we also have some injections above the knee in the hope that we can improve the geniculate collaterals to the infrapopliteal vessels. We're trying to get more information about how cell therapies work. And in addition to getting functional performance, a peak walking time on treadmill exercise testing, which is the standard primary endpoint for peripheral arterial disease, we're getting some imaging studies to assess the collateral vessels, to assess the flow and the tissue perfusion using MR methodologies that have not been previously employed in this kind of a stem cell therapy in peripheral arterial disease. So we hope to get more information about mechanisms of action for any benefit that we hope to see. So I mentioned there's a lot of unanswered questions that remain. Hopefully the PACE study will answer a few of these, but I'm going to go off the reservation for a moment. I'm going to go off the reservation for a moment and ask the question, is stem cell therapy really the right approach? What is the future of our regenerative medicine for peripheral arterial disease? And I'd like to introduce a new topic to you, and that is using small molecules for therapeutic transdifferentiation. And I'm not suggesting to you that we're going to regrow the limbs of our patients with critical limb ischemia, but there certainly are primordial mechanisms for tissue repair in lower vertebrates that are more active than they are in adult humans. I would submit to you that those pathways are present, though, in human fetuses, in neonates. There's demonstrable evidence for that. For example, our neonatal surgeons, when they do intrauterine operations on children for life-threatening congenital heart disease, those children are born without a scar on their chest. They've healed without scar, they've healed with tissue. So much like lower vertebrates that heal with tissue, I think that these pathways may be reawakened in humans. And just to provide some strong scientific grounding for that statement, I submit this paper that we published in Cell in 2012, where we showed that Yamanaka had it half right. The iPSCs certainly a fundamental change in landmark observation, worthy of the Nobel Prize, but there's a lot more to learn about iPSCs, and one of the things that we stumbled across, I think that's very fundamental, is that you can't get nuclear reprogramming to pluripotency without activation of innate immunity. Turns out that the retroviral vector itself that Yamanaka used to create iPSCs activates innate immunity, and that is a critical signaling pathway involved in inducing pluripotency. The Yamanaka factors, ZOC4, ZOC2, KLF4, c-MYC, those are critical for organizing the epigenetic machinery. But the epigenetic machinery is mobilized by activation of innate immune signaling. What we found in this paper published in Cell was that activation of innate immunity either by the retroviral vector or modified messenger RNA or a synthetic double-stranded RNA activates this innate immune signaling that causes global changes in epigenetic modifiers that lead to epigenetic plasticity. There's downregulation of the HDAC family, upregulation of the histone acetyltransferases, and basically the chromatin gets placed into an open probability state that allows for epigenetic plasticity and phenotypic fluidity of cells. Now, once we understood that process, we decided that we were going to apply this for therapeutic transdifferentiation. So other investigators have shown you that it's possible to change a somatic cell directly into another somatic cell, bypassing any need for a pluripotent stem cell. Other groups have done that by using, again, retroviral constructs, encoding transcriptional factors that are important in lineage. I would submit to you that they were doing the same thing, activating innate immunity and then providing the transcription factors that allow for directed differentiation from one somatic cell to another. But we wanted to do this entirely with small molecules, avoid viral vectors and avoid transcription factors. And the way we did this was mimic the retroviral vector with poly-IC, polyinosinic cytidyllic acid. It's an immune adjuvant. It's a double-stranded RNA that stimulates innate immunity, and it's actually approved for clinical use. And then, in addition to that innate immune activator, we provided cues, environmental cues, basically outside-in signaling, factors such as VEGF, vascular endothelial growth factor, and a few other factors. It's now published in Circulation, just came out 2 weeks ago. And the protocol is shown. There's an induction phase where we take human fibroblasts and we introduce poly-IC in the induction phase to increase epigenetic plasticity. Then we provide the external cues, growth factors, BMP4, VEGF, and FGF in the right concentrations, 8-bromocyclic GMP. Later we add a TGF-beta receptor inhibitor to prevent the reverse reaction, mesenchymal to endothelial transdifferentiation. And you can see the FACS analysis shows that in the middle panel, we were successful using this protocol in getting a small yield of endothelial cells. But it's proof of concept. We were able to get 2% endothelial cells from the fibroblasts, the human fibroblasts that went through this protocol. I can tell you now that we've, in the process of getting this paper published, in the year it took us actually 2 years it took us to get this published, we've now improved the protocol, and those results will be forthcoming. We now have a 15% yield, and we're getting into a range with our small molecule cocktail that may be clinically meaningful. But in any event, you can see in the first panel that if we add all the growth factors and all the cues, but we don't add poly-IC, if we don't if we don't activate innate immunity, we get nothing. We don't get any transformation. So the external cues are important. The growth factors, endothelial growth factor medium is important, but activation of innate immunity is required. And I won't go into too much detail. This is published now, as I said, a couple weeks ago in Circulation. But you can see a heat map on the left, which compares the parental fibroblasts to the induced endothelial cells in the two middle columns, and genuine human microvascular endothelial cells on the far right, and of that heat map. And you can see that the IECs, the induced endothelial cells, resemble the genuine human microvascular endothelial cell, not the parental fibroblast. And you can see in the upper panels there that these induced endothelial cells stain for the right immunohistochemical markings. They form vascular networks in matrigel. And in vivo, when we we put human fibroblasts into matrigel with the cocktail, we can get self-assembly of these induced endothelial cells into capillaries that conduct blood flow. And some other studies, functional studies down below, showing that they can make nitric oxide and angiogenic cytokines. So I'm going to conclude with this slide just to get you thinking about this concept that we call transflammation. And the idea is that when a cell is confronted by an injury, when it's confronted by damage or a pathogen, that damage presents damage-associated molecular patterns. Those pathogens present pathogen-associated molecular patterns that trigger receptors on any cell to activate innate immunity. These pathways are well known, well described by immunologists, but what was not known and what we stumbled upon was that activation of innate immunity causes global changes in epigenetic modifiers, as I mentioned to you, that put the chromatin into an open probability state that allows the cell to be therapeutically manipulated. So what are the potential applications we're thinking of? Well, in a situation where you have an injury like a myocardial infarction and you have cardiac fibroblasts migrating into that area of injury, a small molecule cocktail could potentially transdifferentiate some of those cardiac fibroblasts and endothelial cells that would provide the microvasculature, the nutrition, and the niche for true tissue repair. So this is the crew that's helping me at our Center for Cardiovascular Regeneration at Houston Methodist, and I thank you for your attention.
Thank you very much. I have the distinct honor of introducing an icon of cardiovascular medicine, Dr. James T. Willerson. He is the president and director of the Texas Heart Institute. He is the Alkek Williams Distinguished Professor and holds the Edward Randall III Chair in Internal Medicine. He is a former editor of Circulation and a clinical trialist who has led the field in preclinical and clinical studies of cardiovascular stem cell research.
Too generous an introduction, but I appreciate it. I grew up in San Antonio, so it's nice to be back here. Brings back many fond memories, and I thought Dr. Cooke's talk was terrific. We had established between the Texas Heart Institute and the Hospital Procardico in Rio de Janeiro, Brazil, in 1996, a clinical research relationship. That's a heart hospital in Rio. I thought we would probably begin some clinical trials there with gene therapy. But we've been also involved with some animal models with experimentally created infarcts and using stem cells. And had noticed an interesting process known as nuclear fusion, and also seen benefit from stem cells in the animal models and no harm. So Dr. Emerson Perrin, who sends his regrets for not being here today, some family issues kept him from coming. And I and others were able to persuade the Brazilian government that year 2000 or 2001 to engage in treatment of patients with ischemic cardiomyopathies, large dilated, often basketball-sized hearts, just quivering at the top, treat them with their own bone marrow-derived stem cells. These are patients who had no other option for treatment. And we went through about 10 Brazilian committees to get permission. We were allowed to treat 4 patients for safety originally. And we treated a total of 14 who had 7 match controls. This was the first such study in the world in injecting bone marrow-derived stem cells directly into human hearts in patients with ischemic cardiomyopathies. It's published in Circulation 2003. A mixture of Texas Heart Institute and Brazilian cardiologist, Dr. Perrin, the lead author on it. And what we showed was this not controlled, open trial was an improvement in the mobile function of the heart, an improvement in blood flow, and double velocity, regional wall motion at sites where we inject cells. There's a catheter that we used for such experience with this matters too. No good catheter. It's put into a peripheral artery, femoral artery, usually threaded backward into the aorta across the aortic valve. And once into the interior of the left ventricle, where the tip of that catheter touches LV subendocardium, it measures electrical potential and also the velocity of wall motion. Has a needle on the end of it that we can protrude and withdraw, so we can inject cells at sites that we believe are reversibly injured based on the electrical potential. Not scar initially, and certainly not normal areas. This slide is representative of what we found in this very small, not placebo-controlled trial. Top 2 panels are nuclear spec studies. The bottom 2 panels are functional maps. The black dots on your bottom left are where we injected this patient's heart. Let's go back to the top 2 panels. On your left, top and bottom, are pre-treatment pictures. And then on the top right and bottom right are first, the blood flow study is 2 months after cell treatment, and the functional study is 4 months after cell treatment. The black color on your left is putative scar in this human heart, and the white colors are stress-induced ischemia. Remember, these patients have ischemic cardiomyopathy. Within 2 months, as you see left to right, top panels, there's a reduction in, quote, scar, and a reduction in some, but not all, of the stress-induced ischemia. We'll go down to the bottom 2 panels, And red is bad, bottom left. Red is dead, or almost. And then moving 4 months later, bottom right, you see the improvement in some of the areas that we injected. Now, we were, of course, curious about why we got this beneficial effect. And I mentioned we'd done some experimental studies, but this is one I want to call your attention to. It's published in Circulation in 2003. And what we did was take skid mice. These are mice that are genetically engineered so they cannot reject micelles. I'm not sure they'd want them, but human progenitor, youthful cells injected into the tail veins of these skid mice in whom we had created an infarct by ligating the LAD. By the way, the mice don't seem to mind this. They run around their cage as if nothing's ever happened to them. And then with a little experience, virtually all of them survive. So we injected our human endothelial progenitor cells into the skid mouse with an infarct. Using this model, you can unambiguously determine where the cells go and what they do when they go there because of the differences in the X chromosome between the mouse and the human, as well as using certain markers for myocytes. And what we found was that the injected cells went to the region around the infarct. They clearly differentiated. These are largely CD34 positive cells. They clearly differentiated into smooth muscle cells and endothelial cells and a few new myocytes. But most of them fused their nucleus, human cell nucleus, to the mouse cell nucleus in reversibly injured cells, not dead cells and not normal cells. What's the point of that fusion? Shown here, this is published, I think, in Circ Research about 2007, but the references are at the bottom of the slide. In order to look at the meaning of nuclear fusion, we co-cultured the skid mouse myocytes and the human cells. And then we're able to manipulate the environment to see if we could influence the fusion phenomenon. And it turns out that if you make that environment hypoxic, you increase fusion. If you introduce interleukin-6, you increase nuclear fusion. If you introduce TNF-alpha, you increase the nuclear fusion. And if you do all three of those things, the environment's hypoxic in both of these cytokines, nuclear fusion increases by 11-fold. Still, it's at a low level. And what happens with nuclear fusion is it turns on the cell cycle. It stimulates cyclin B1 activity in the fused nuclear cells and bromodioxyuridine uptake, and it results in the generation of new myocytes. So here's a way to create stem cells without killing anything, without injecting anything. Nature's way, our maker's way of generating new cells. Bad news, it occurs at a low level, not enough to really help a severely injured human heart. But in the course of this work, we also identified the receptor for nuclear fusion. It's an alpha-4 beta-1 BCAM receptor. If that could be overexpressed, one might amplify this phenomenon. This is also the vascular receptor for inflammation. And we have agonists and antagonists to it now and are proceeding to try to identify in vivo vascular inflammation with this help. I'm going to skip through the evidence for what I just told you. Otherwise, you and I will be here all day. But go to the top right-hand panel with me. In these studies, here we caught a nucleus in division. And you see green and red colors in both halves of that nucleus that's dividing. Those are fused nuclear and murine nuclei in both, spontaneously dividing. One of the questions that's come up in this kind of work is when you inject stem cells into the heart, how long do they last? And the answer is it depends on the cell. In this study, using the same model, SCID mouse, and injecting progenitor cells, CD34 positive cells, either into the tail vein or directly into the left ventricles in the infarcted mouse heart, and using bioluminescence, PET imaging, and MRI, shown here, top panels. Your very top one on the left is prior to injection. Then after that, you can see the time markers and the increase in the first week in the presence of these cells as if they might be activating and increasing in number. And then down through the bottom panels, we followed these mice for 52 weeks. And you see there's a gradual decline in the number of cells. Go with me to the graph on the bottom. You can see that decline. But for the CD34 positive cells, some are still there a year later. If I showed you the same experiment with mesenchymal cells, they'd all be gone in 2 weeks. So the cell type makes a difference. But the functional effect persists for both. Here it's shown for the CD34 positive cells. Let's go to the bottom graph. In the red color are the treated mouse hearts with the CD34 positive cells. We're measuring ejection fraction. And you can see an improvement in the ejection fraction beginning about the same time that there seemed to be more density to the cells in the human heart and lasting for as long as we were willing to measure it, which was 25 weeks. Mesenchymal cells do exactly the same thing, even though they're gone in 2 weeks. So we need to understand that. One possibility is that the mesenchymal cells upregulate the number of c-kit cells in an animal heart. And that may be the explanation, or a part of the explanation, for the persistent effect of the mesenchymal cells. For the CD34 cells, this positive effect on ejection fraction is a blood flow effect. It can be blocked by an antibody to VEGF, but not blocked by an antibody to the vascular receptor for inflammation. In 2004, Dr. Perrin and I and our colleagues went to the FDA in this country. We wanted to do a study like the one we'd done in Brazil, but this time randomized, placebo-controlled, and blinded at the Texas Heart Institute in Houston. These same patients with ischemic cardiomyopathies. There were about 60 people in the room. It may have been my false impression, but I thought they seemed a little embarrassed about our presence there. Maybe embarrassed because we went to Brazil to do the work originally. Nevertheless, they wanted to discount the work in Brazil. They didn't believe it. And after about an hour and a half of questions, which were very difficult, I thought to myself, you were right the first time. They're not going to let you do this work in the United States, even though these are human stem cells. And we've already shown the safety for them. And by that time, some others had too, particularly two German groups, Zires and Strauer. And I said to them, listen to me. I'm an American, I'm a Texan, I'm an adopted Houstonian, and I want to tell you something. If you don't let us do this work in Houston, you're going to watch it done all over the world, because we are going to do it. There's a long pause. I thought maybe I'm on my way to Leavenworth. But the questions got a little softer, but it was still a year before they let us begin this trial. And this would be a small trial, 30 patients, 20 treated, NOGA catheter injections into the heart of these patients with ischemic cardiomyopathies, 10 controlled with placebo and blinded. We wanted to show safety and we wanted to look for a signal for efficacy. This is published in the American Heart Journal, and I think the references is down at the bottom several years ago now. What we found was overall no improvement in ejection fraction or reduction in systolic volume or improvement in blood flow. And it really left us scratching our heads about what could be wrong. Fortunately, we had saved a sample of the aliquot we injected. We wanted to look at the normalcy of the cells that we injected, or lack thereof. This slide shows you, if you look at that bar across the center, that's what mesenchymal cells ought to be able to do in colony-forming units in culture. In our treated and controlled patients, there were only 2 patients in the entire study whose mesenchymal cells had normal colony-forming capability. So these cells were dysfunctional. And we looked at a number of variables, but the one that leaped out at us was age. John talked a few minutes ago about as we get older and as we develop various diseases, stem cells are dysfunctional. And they are, and I actually think that aging is a stem cell deficiency by and large. And we're working on that. But here in this study, if we looked at the colony-forming capability of the mesenchymal cells and those under 60, shown on your left in the yellow bar, and those over 60, you can see the difference. The younger cells were functionally normal and the older ones were not. So then if we took the primary endpoint of this study, which was maximal oxygen consumption, and we looked at those under 60 as controls or treated, you can see that there was a significant benefit, and rather significant, between the original value in a patient and the 6-month value, or I think it's 3 months here, but it was the same as 6 months. Very significant increase in maximal oxygen consumption. So we've got to pay attention to this. The cells do matter. Now, more recently, we've had the privilege of engaging in the CCTRN with the largest autologous bone marrow-derived stem cell therapy in humans with ischemic cardiomyopathies done yet. 92 patients. And this was published in JAMA a year and a half ago. And basically we showed what we had showed in the randomized trial in Houston. Overall, in the treated patients, and we used 100 million cells here, there was no improvement in end-systolic volume or remodeling variables. But if we looked at ejection fraction, shown on the left, in the treated patients, there was a small and statistically significant increase in ejection fraction, about 3%, I think. Very similar to what Zaire in Germany has been showing in patients with acute infarcts treated with bone marrow-derived stem cells. The bone marrow-derived stem cells, if they're functionally normal, will generally have a positive but very small effect on global or segmental function. The control patients are shown on your right, and you can see that a number of them deteriorated. However, it gets a little more interesting. If you stop and think about this, we're from 92 patients, or the cohort that are going to be treated with cells, we're taking what's in their bone marrow, and it's not going to be the same for every patient. It's not like giving 0.5 milligram of some medication. They're highly variable. So what happens if you look at the bone marrow phenotype from control and relate it to the improvements in ejection fraction, Dr. Willerson? Well, that's shown here, where there were increases in CD34 and CD133 positive cells in the bone marrows of these treated patients. There were additional and substantive increases in ejection fraction. So much so that for the CD133 positive cells, where they were increased in number in the bone marrow, there was a 5.96% greater absolute unit increase in LVEF. So what one's injecting does make a difference, not only whether they're old, but what is in that bone marrow. Certainly not a surprise. One stopped and thought about it. So we've been a little more demanding recently. What if you looked at a triple endpoint that was positive in function in this trial of 92 patients. That is, what if you looked at the bone marrows in the patients who increased their ejection fraction, decreased their end-systolic volume, and had an increase in maximal oxygen consumption versus those that didn't? What would the bone marrow look like? Well, it turns out that there's a larger number of B cells and a larger number of CRCX4 cells in the bone marrows of patients who have positive responses to that triple endpoint. And by the way, that would be about as demanding a requirement for a positive clinical trial as you could exert from just the functional variables. There was also a decrease in the number of colony endothelial forming cells and monocyte macrophages in these bone marrows. Now we're trying to publish this right now. Hope it's not going to take us 3 years, John, but it might, given the climate. And we want to test this prospectively. So in a future trial that we're hopefully soon to launch in CCTRN, combining c-Kit cells and mesenchymal cells in these same patients and looking at them individually, we'll challenge this observation. Does it really predict the magnitude of response? Now, very quickly, I want to just mention the c-Kit cells and the cardiosphere-derived cells. Has that been discussed in this conference already? No. Okay. I'm going to be quick. The heart and every organ of the body has resident stem cells. In the heart, the one that certainly looks promising at the moment, although there's a lot of controversy about it, are the c-Kit cells. And Dr. Anversa, currently at the Brigham, and Dr. Bowley in Louisville have been very interested in studying these cells in human hearts. And I'm going to show you a trial they've done recently. And then Dr. Marban and his colleagues, while he was at Johns Hopkins and now moved to Cedars-Sinai in Los Angeles, have identified a group of stem cells that they refer to as cardiosphere-derived stem cells because they round up in culture. When they culture segments of the myocardium in which these cells are present, they're clusters of cells. These are not homogeneous stem cells, the cardiospheres. They're a mixture of the kinds of stem cells you've been discussing throughout this conference, but CD34, mesenchymal, c-Kit, CD133, and so on. This is the Scipio trial, Dr. Bowley's trial, and in this case, the patients had had ischemic heart damage, and they were to undergo coronary bypass surgery. But Bowley and his colleagues took samples from the right heart in these patients to separate out c-Kit cells and expand them in culture. The patients had cabbage procedures, and then those that would be treated about 4 months after their cabbage had the c-Kit cells injected into an infarct-related artery. And there were controls. Now, the follow-up of these patients continues now, but look at the top left-hand panel there, and on the right side of that, those are ejection fractions, and you see immediately that the increase is greater than what I've shown you or alluded to for what bone marrow-derived cells do. And then if you look a little later at a year, top right, that increase is continuing. So this is very encouraging and certainly something we want to build on. This is Marban's study with the cardiospheres. And what he did was take patients with large infarcts, take endocardial biopsies, isolate c-Kit cells from these biopsies, grow them up in culture, expand them, and several weeks later, treat the patient in the infarct-related artery or a control. And what he found primarily was a reduction in infarct size. Go to the bottom part of that slide, or a reduction in scar size. Now I tell you this a little tongue-in-cheek, because the gadolinium MRI enhancement identification of scar is probably more than scar. scar and inflammation and edema. But there's clearly a reduction in this trial on the left-hand bottom panels, then as you move to the right over time, and it's significant. And there was also an increase in regional contractility in the treated patients. We've been looking at allergenic mesenchymal cells, study led by Emerson Perrin and help from some of the rest of us. These are patients with ischemic cardiomyopathy, but trying to get away from the fact that aging cells become dysfunctional. We've taken mesenchymal cells from one donor, a 19-year-old donor. These are bone marrow derived, and one removes the most primitive mesenchymal cells by an immune absorption technique. From that one donor, we treated 45 humans with ischemic cardiomyopathy, no catheter injections, and had 15 controls. What we found was that in the highest dose that we used, namely 150 million cells, there was improvement in function, improvement in flow to the heart, but most importantly, and if you can read this down about 2/3 away toward the bottom, there was a marked reduction in major adverse cardiac events going forward, major reduction in mortality, major reduction in rehospitalization, and an improvement in quality of life and functional class. We're also trying to publish this right now. But we have published this one. I think we're up to the PRECISE trial. We've taken autologous mesenchymal cells from adipose tissue in another relatively small trial and injected the adipose tissue-derived stem cells. These are largely mesenchymal cells, but not completely, into the human heart, and found the same benefit that we found with the Mesoblast trial I just described to you, improvements in function and flow and stabilization of clinical course. Dr. Hare, who's been here and spoken at this meeting, has looked at allergenic and autologous mesenchymal cells in humans, and this is from his work, and shown that they exert about the same effect in the human heart with ischemic cardiomyopathy. In both our trials and Dr. Hare's, there's no real evidence of immune rejection of the allogeneic cells. There's a transient increase in an HLA marker, 10% of patients in our trial, which was not expressed clinically in any manner and disappeared over time. So the bottom line of this is it looks like one may be able to use youthful mesenchymal cells, not have them rejected in older hearts, and get around this business of the aging cell. Josh Hare has also looked at the combined effect of c-Kit cells and mesenchymal cells in a porcine model of myocardial infarction. And this is from his work. And the bottom line of that is that the combination of the cells decreases infarct size in this pig model twice as much as either cell type alone, and again appeared safe. And this is part of the basis for our concert trial with c-Kit and mesenchymal cells. But we have multiple stem cell types. That's not by accident. They've all got to have some different or additive function. And my bet with you right now is that combinations of cells, the right cells, will exert bigger effects than single cells. Hare's work certainly would be in support of that. Okay. We have taken aging mouse cells, C57 black mice and ApoE deficient mice who have vascular disease. And we have shown that the overexpression of telomerase and myocardin, but not either one alone, rejuvenates the aging mouse cells back to youthful function, both in culture and in vivo. And in the in vivo shown here in the very bottom panels, if you look at the blood flow in this hind limb ischemic model in mice on your far left, which is normal, and then the decrease that occurs when you ligate femoral artery, and then the restoration of flow on your far right in aged cells now made youthful by the overexpression of telomerase and myocardium. John alluded to work of this type too, but I want to mention work from California done by Mark Sussman with PIM1 kinase. He's shown that the overexpression of PIM1 kinase also restores these cells, human cells actually, to a more youthful function. And Anversa has done the same thing with stem cell factor in mouse cells. So there are going to be ways to rejuvenate the AIDS cell. What I don't know is how stable that rejuvenation is and how safe it is. And we have to find that out. We, some years ago now, led by Dr. Robert Schwartz at our institution, have converted human skin fibroblasts into contracting heart muscle cells. This is published in PNAS about 2 years ago. But within 2 weeks, we could take a biopsy from your hand and convert some of your fibroblasts into your contracting heart muscle cells. We've built on that. I'm not going to say much more about it because it's not published or completely protected yet by patent. But there are other cell types that are even more robust. This is done with 2 transcription factors, which are shown in the title. This is our team, which I'm very proud of. Dr. Perrin, Dr. Taylor, Dr. Schwartz, Dr. Martin, Dr. Dixon, Dr. Cabrera, Dr. Silva, and an older person who's part of the group. And some of our technical help is right here too, and you all know them. We're very proud of them. This is where we work. We're right next door to Dr. Cooke's Research Institute, which is magnificent itself. But that's a Texas Heart Institute, and both of these right across the street from the MD Anderson in Houston. If you're there, come and see us. Thank you very much.
Thank you all. So I'm going to open up the forum for questions. As we just learned, these stem cells in the cardiovascular disease area do have the power to transform it. So whoever has questions right now for our speakers? Any questions? I have a question. So which cell is going to be the best cell to cure cardiovascular disease? Dr. Willerson. Hello?
Darcy, you know very well. Can everybody hear me?
No.
It's being provocative, but you know very well we don't know. She's being provocative, but we don't know yet. And that's exactly what we're trying to find out. You've already heard my prediction. It's going to be combinations of cells. Not going to be one cell, but a combination. And it may ultimately include some cells that we have difficulty working with. But it's really been wonderful to discover the potency of normally functioning human adult stem cells. And I think it'll be combinations of. It may not just be 2 cells, it may be 3 or 4 that do different things. Our goal is a regeneration of the whole heart. That's what we're working on. And I know Dr. Taylor discussed that. So at the moment, this is a work in progress. It's patchy, it's additive, but that's where we hope to get. Is that possible? Of course it is. We're all products of stem cells. The union of our father's sperm and mother's egg generated billions of stem cells in the blastocyst. I believe ordained by our Maker to be your heart, your kidney, your spirit, your intellect, your appearance, every single thing about you, through genetic signaling, some of which we're beginning to discover. So if that's correct, then the only issue is, you could ask me, are we smart enough? It's not, can it be done? It can be done. And I really believe every organ in the body will be regenerated from stem cells, small molecules, and additive factors.
Thank you. Question?
I guess I can shout. Well, I better probably make a better impression about the FDA. I work with the FDA. My specialty is oncology, and I'm so glad to have this opportunity to listen to your presentation. Excellent. And apologize if we have given you some hard time before, even though I wasn't there. But
From the FDA.
I would like to, one question is just thank you so much for your excellent work and the presentation. I would like to ask you, first of all, if there is any other or more obstacles and problems in your dealing with FDA. I would like to take that back to my colleagues and my upper management. There is a room for us to improve. So that's my first question.
So we don't have enough time. We don't have enough time to address that question fully. But I would say that one major change that I think should take place is that we should allow therapeutics to go out after phase 2B, particularly for diseases like critical limb ischemia, where we don't have good solutions. After phase 2B, if something shows a signal of efficacy and it appears to be safe, let it go out. Let it go out, and then whatever the company is that's responsible for this, they have to come back to you 5-6 years later with the data. So it would be post-marketing surveillance. And it would go out with a black box warning that it hasn't been fully tested yet in definitive trials. So you let things go out after phase 2B. It would get therapeutics to the market much faster, get therapeutics to our patients much faster. There's greater risk. People would be warned. They would have a black box warning. And then the pharmaceutical company, biotech company, would have to come back 5 years later with the definitive studies, with the definitive phase 3 studies. And then that would allow more general marketing. So during that phase 2B process, the marketing would be very restricted, would be to specific patients that are in need, and that would allow us to get therapy to our patients much faster. So I think that's a major change in thinking that could really accelerate drug development.
Can she do her second question real quick?
Or maybe comments. That's just right on the target. I would like to talk to you more after this, off-site, but what I want to let you know is that FDA started this by law is a breakthrough therapy designation request. So exactly fit your situation. If you have promising data in the phase 2, you will apply for the breakthrough therapy. We will fast lane to your development. You have absolutely an upper, middle, and lower management. We right away work on that. So I will talk to you more about that.
One last question.
So thank you for wonderful presentations. I just want to pick up on that last concept that you articulated about having sort of a bifurcation after phase 2. You know, Andy Grove made a keynote speech, I don't know, 2 or 3 years ago in Pasadena to the World Stem Cell Summit. And in his address, that's exactly what he was proposing. You know, at the time, there seemed to be a lot of issues, everything from how to handle the statistical aspects of having that kind of bifurcation with follow-on phase 3 trials, plus the expense of manufacturing and processing that bifurcation. And I'm just shortlisting all of the issues, but it's interesting to hear you, 2 or 3 years later after Andy Grove addressed this summit, saying the same thing. And maybe if we're serious about that, it really needs to be explored in a very systematic way.
I'll say one thing that I'd like to hear Dr. Willerson's comment on this, but I'm a physician, as is Dr. Willerson, and our oath is to first do no harm. So that is always the concern when the regulatory process is accelerated. However, there is harm that can be done by extending the regulatory process to a point where it makes it very, very difficult to get therapies through this regulatory roadmap. Small companies can't do it. And that reduces innovation when small companies can't bring a product to market. Now, there's another thing to say about this in terms of first doing no harm. Because the regulatory hurdles are so high, when you go to phase 3 definitive studies, you have to make certain that you do a patient selection that will give you the desired result. And when I've done about 24 clinical trials for pharmaceutical companies, the inclusion and exclusion criteria are so rigorous that only 1 out of every 10 patients that has the disease actually gets tested in that study. And then what happens is the drug gets approved. And then everyone gets it. The other 9 out of 10 get it. And you never end up seeing in those initial phase 3 trials potential side effects. So I think that what I've proposed, or what Andy Grove apparently has proposed and others are talking about, is a regulatory roadmap that is shorter, that'll get therapies to market quicker, but will still be regulatory oversight. Jim, what do you think?
Thank you for your question. I agree with Dr. Cooke completely, but I want to take one additional view. So heart disease is the #1 killer of men, women, and children in our country and throughout the world, every major country in the world. Women aren't very aware of this, but more women die of heart disease than of all cancers combined. If you're a woman, you're 5 times more likely to die of a heart attack than of breast cancer. And 1 out of 2 men worldwide have heart and vascular disease during their lives. The most common heart disease is heart failure. And patients with end-stage heart failure can't walk from here to the ground because they don't have the breath or the energy to do it. Many of them can't sleep lying down. They sit up if they can breathe at all. And 2/3 of them are dead in 4 or 5 years. So we transplant hearts, somebody else's heart into them with all the consequences of that, and still not a very good 10-year survival. And we use mechanical devices, which have gotten quite good. But they're still mechanical devices and dependent on energy source and not perfect. Wouldn't it be nice to be able to regenerate enough of the heart or other organ that's injured to not require somebody else's organ or a mechanical device? Do no harm. That addresses the intention of us and all of us who work in these fields, not to harm somebody directly. But I think the do no harm is more expansive than that. I think do no harm means help. Help beyond what can be helped now and do it as quickly as possible. There are millions of people all over the world with heart failure. And in many places, they don't even have access to heart transplants or LVADs, probably never will. So that's the other side of do no harm, I think. Thank you for your thoughtful question.
The session's concluded. Thank you very much.